Welcome back, dear student, and good morning. I am Dr. Savita Tidame, and we are studying the chapter number 3, that is a plasma membrane. I welcome you all in the online lecture series. Okay, so let's take a brief recap of this chapter, what we have studied up till now. So during first 3 lectures, we have studied the physical, that is composition of the plasma membrane, right? Protein, lipid, carbohydrate, their percentage, types that we already studied. Then worldwide accepted fluid mosaic model of the plasma membrane we have studied. Even after that, we have studied the transport across the membrane, right? How the biomolecules from cell outside, how it get entered inside the cell, or even from the cell inside, how the molecules, excretory material, toxic material, they have been exported outside or they have been exported through the cell. So that is again further divided into simple diffusion or simple diffusion, further simple and facilitated diffusion, right? And even active transport, right? So passive transport comprises simple diffusion and facilitated diffusion, and second type is the active transport across the membrane. So that we have studied in detail in the previous 3 lectures. Now today we'll be studying the process of pinocytosis and phagocytosis, that is also kind of a import across the membrane. So that is called as endocytosis. So suppose any material which is taken from cell outside to the cell inside through the plasma membrane, that is called as a endocytosis or inward movement of the molecule. Now, why the endocytosis is further get divided into 2 types, that is pinocytosis and phagocytosis, right? Pinocytosis and phagocytosis. So, pinocytosis is what? Whenever the liquid molecules or the solid molecules, they are taken inside by the cell, that is called as a pinocytosis. And the pinocytosis, dear students, it is also called as the cellular drinking. Right? Pinocytosis is also called as a cellular drinking, whereas phagocytosis, it is a process in which the solid particles are taken inside by the same invasion of the plasma membrane. So this is called as a cellular eating. What phagocytosis is called as cellular eating. Now let's see the procedure of pinocytosis and phagocytosis with the help of this diagram. Okay. Alright, so whenever the intake of a fluid material, here is a solid droplet I have shown. Solid itself is what? Solid is nothing but a fluid material into the cell by formation of a pinosome. So pinosome or pinocytic vesicle is called as a pinocytosis, right? Intake of a solid molecule or the fluid material inside the cell through the formation of a pinosome or a pinocytic vesicle, that is called as the pinocytosis. And that has been, this procedure was described by the scientist Lewis in 1931. You can imagine, dear students, in 1931 this was invented by the scientist called, or his name is Lewis. Okay, then after that, it's a kind of, that is it's a kind of cellular drinking by the cell, and this mode of intake of nutrient has been demonstrated in the endothelial cells and in the erythroblast. So in the blood cells, blood cellular linings, as well as in the W.B.C.S, that is one of the blood cell in which a blood cell type, okay, RBCs, erythrocyte means RBCs and leukocyte means WBC. So in the erythrocyte, that is in RBCs, such a kind of a pinocytosis is often observed. So fluid in the fluid molecules intake with the plasma membrane move through narrow channel formed a temporary as deep invasion into the cell interior. You can see here this solid molecule when comes in contact with the plasma membrane. So plasma membrane going to detect it immediately, and that detection or close contact of that solid molecule in the or along with the plasma membrane start or trigger the invagination of the plasma membrane. So whenever the plasma membrane it get invaginated, that solid molecule is taken inside that invagination, and slowly it grows further, and then it get pinched off. The remaining membrane is get fused together, and the pinched off vesicle is now ready. It is detached from the plasma membrane. That is called as a pinosome. What is called pinosome? Because it is a pinocytic vesicle. No, so it is called as a pinosome. Now this pinosome, where does it go further? Yes, it is not going to use as it is, but what happened? This pinosome, dear students, further is get fused with the lysosome. Okay, because itself it is not able to digest, no? And for the digestion of this protein or whatever material is there, liquid material is there, lysosomes are utilized. So it get fused with the lysosome. Further, the nutrient they are released into the cytoplasm, right? And as it is a liquid one, there is no undigested material is going to left. So whatever nutrients are there which set free after digestion with the primary lysosome, and then the nutrients are utilized by the cell for its metabolic activity. So this is the simple procedure of the pinocytosis. And this procedure of the pinocytosis is said to be mediated by some kind of a inducer mechanism. As we said that the liquid molecules or the solid molecule, when comes in contact with the plasma membrane, it going to trigger the membrane. So that trigger or some mechanism or inducer is there which trigger the formation of the invagination curve, right? So that trigger mechanism is there, and number of triggering molecules are involved. Literally, if you see, number of triggering molecules are involved. So out of that number of triggering molecules, protein in solution or protein is one of the good inducer. If the solid particles are having traces of the protein, or they are made up of a protein, or they are proteinous in nature, then what happens? This invagination will be faster. So protein is one of the good inducer that is going to trigger the invagination as compared to the DNA, RNA and fatty acid, right? As compared to DNA, RNA and fatty acid. So DNA, RNA and fatty acid, when comes in contact with the plasma membrane, their invagination is again very slow, no doubt. It doesn't mean they are not going to trigger the invagination. They will trigger the invagination, but the invagination process will be very slow as compared to protein, because the protein is one of the very good inducer for pinocytosis, isn't it? So all substances surrounding the cell membranes may not be engulfed. We know that number of substances are present around the cell. Excretory material is also there, inorganic substances are there, liquid material is there, right? Number of substances are present around the cell. It doesn't mean all those substances are need to be imported or need to be taken inside by the cell. No, it doesn't mean like that. All the substances present around the cell may not be engulfed. Hence, the process is what? Highly selective. So, the process of a pinocytosis is highly selective. So, the surface of plasma membrane has a specific receptor. How this selectivity is there, and how the plasma membrane decide which material is need to be taken inside the cell and which material is not taken or it is rejected, right? How it is decided? Yes, it is decided on the basis of a receptor molecules. If you see, plasma membrane is surrounded by means of a number of receptors, and the receptors, they are very specific, just like a lock and key mechanism. A specific lock can open with a specific key, right? So here is also the specific receptors are there for a specific kind of a biomolecule. Any biomolecule cannot attach with any receptor, right? So specific kind of receptors are there which only allows the entry of certain molecules which get bounded or attached to it and carried through the invagination. So endothelial cell linings, that is cellular linings of the capillaries and blood vessels, may be cited to prove this point. So through the blood vessels, molecules or selective we can say the transport of a certain molecules taken so from the blood vessel nutrient. Okay, let me explain this with another example. Whatever food we eat, where it goes to the stomach where it digested, and further it goes to intestine where it is absorbed, right? And after absorption, what happened? After absorption, nutrients are entered into the blood, and from the blood, circulating blood, along with the circulation, the nutrient are transported to the cellular level, to every cell of our body gets that nutrition from the food we eat, right? And how the food material is being transported, nutrients are transported to the cell line, because it is not open, no, everything is not open, alimentary canal is completely closed, right? Even blood cells, blood vessels, they are also intact. Then how the material nutrients they are transported? Think. Yes, they are transported through the process of a pinocytosis, phagocytosis, right? So digested material, if it is in the form of a liquid like this, they are transported through the pinocytosis. So pinocytosis is very well studied in the blood cell linings through which the nutrients are transported to cells, right? So pinocytosis is very well studied in the blood cell linings. Okay, so materials from the bloods are transported into the interstitial spaces through the small vesicles that are formed to the cell border, and these vesicles loaded with the nutrients are then transported to the other side of the cell where they fuse with the cell membrane and discharge the content outside. So the pinocytosis is one of the endocytic procedure taken place across the plasma membrane of the cell whenever they want to, or whenever the liquid molecules or solute molecules they are taken inside by the cell. Okay, now after that, dear students, let's move ahead and study the phagocytosis procedure. Phagocytosis. Okay, so engulfment, as I told you earlier, so phagocytosis is what? It involves the intake of a solid food particle, right? It involves the intake of a solid food particle. So engulfment of a solid particles with the help of a plasma membrane has been observed in varieties of different cells, such as the leukocytes. Yes, in our body, if any kind of a infection is there, right? If you got some infection, if you got some injury, so that injury is healed, it is covered by it get covered within few days. How? Because our soldiers of our body, that is the WBCs, right, leukocytes, they get ready for the attack on foreign cells as well as bacteria, viruses, whatever foreign material is there at the site of a wound. Even not only that, they also eat up worn out cells. Those cells are getting dead. They are also being completely finished or eaten by the phagocytic cells, that is WBCs. So phagocytosis, that is nothing but engulfment of a solid food particle with the help of a plasma membrane, invagination of the plasma membrane, and this phagocytosis process has been observed in varieties of different types of cells such as leukocytes, endothelial cells, fibrocytes, and splenocytes. So the cells ingest or swallow. Okay, this is cellular ingesting or a swallowing. Swallows of foreign bodies like bacteria, harmful matter, and other inert substances are by the process of phagocytosis, and as it is an intake of a solid food material, so it is called as the solid particle, so it is called as the cellular eating. So phagocytosis literally means the cellular eating, and pinocytosis literally means the cellular drinking. So it is found commonly in the protozoa. Phagocytosis, where it is found? It is found commonly in the protozoa among certain cells of the metazoans. So phagocytosis has been observed in amoeba. For this amoeba is one of the protozoan animal. Okay, and amoeba, here I have shown in the diagram, this amoeba is often showing the phagocytosis for eating, right? It is often showing phagocytosis process for the eating, for getting its nourishment or nutrition, it performs a phagocytosis procedure. Here I have shown, or here in this diagram, what is there? There is a lysosome, nucleus, cytoplasm, plasma membrane of the amoeba, and this is the food particle taken in. Here, the food vesicle is formed by the outward movement of the pseudopodia or formation of a pseudopodia. Pseudopodia is a false legs, we can say. So here the plasma membrane grows around the food particle. It used to engulf it, forms a vesicle, and that vesicle is get detached from the plasma membrane. This is now called as a phagosome. The vesicle formed after ingestion of the food particle or after ingestion of a solid particle is called as a phagosome. Now that phagosome, how the solid material is get digested? It's again a question. Taken simply inside the material is not enough, right? Doesn't mean the cell will get nutrition. It has to digest it further. If we eat whatever food, if you eat the fruits or if you eat puri bhaji, something like that, and it don't get digested, your stomach deny to digest, don't secrete HCl, then how the food is get digested? No, food cannot be get digested further, right? Unless and until the metabolic activity started, stomach lining secretes the HCl as well as other enzymes. Even peristaltic movement is also equally important. Then only digestion takes place and will get the nutrient. So similarly, here in this case also, though the solid material, whatever is there, is taken inside by the amoeba, it's not enough to get the nutrition. Further, that solid material or phagosome has to be digested. So here the lysosomes are there. These lysosomes get attached with the phagosome, forming a secondary lysosome. This is called as a secondary lysosome or digestive vesicle, and inside the digestive vesicle, lysosomal enzyme act on this solid material, okay, and carry out its degradation or digestion, and digested nutrients are then released into the cytoplasm. Yes, digested nutrients are then released into the cytoplasm, and as it is a solid material, so some undigested part will be there, right? Yes or no? Yes, some undigested part will be there. That undigested part, if remain inside the cell, असं होऊ शकतं का? Does it remain inside the cell? So let's correlate with our body. So whatever, every day we used to have the food. In the morning, in the afternoon, in the evening, tonight, every time we used to keep on eating. What happened? It get digested. We get nutrition from that food material, right? And after that, the undigested material, some part of that food is remain undigested. Our body cannot digest it. Yes? So where it goes? It is defecated. It is thrown outside the body in the form of a unwanted or undigested material or in the form of a fecal matter. Right? It has to thrown out. Otherwise, it get accumulate in our stomach, in our intestine, and it get blocked completely. So person cannot digest further, and ultimately the life, or you can say the death, is ultimate. So here in this case also the same kind of situation, the unwanted material or undigested, after digestion of this some part of the solid material is remain undigested, right? And that undigested material is, if remain inside the cell, then here the cellular loading will be there. The cell will be full of that undigested material, and that is not even good for the health of a cell. The cell will die immediately or quickly. Right, so that undigested part of this material, phagosome, is need to be thrown outside the cell. So for that, this vesicle, digestive vesicle, is now containing undigested material or residual material, that is called as a residuosome, and then that residuosome get fused with the plasma membrane, and the waste material is thrown outside. Right, so this is a simple procedure of the phagocytosis. So during the process of phagocytosis, number of events, number of things have been observed. So when there is a material near the phagosome, the cell relays a signal how it get detected by the cell. if something like a food material or solid material is close to us and we have to engulf it, I have to engulf how the cell is deciding. Okay, so some kind of signaling is there, right? Some kind of events must be takes place on the cell membrane, is it? Yes. So when there is a material near the phagocyte, or phagocyte means a cell which carry out a phagocytosis process. So when there is a material near the phagocyte, the cell relays a signal. The cell is going to release some kind of a signal to other cell, right? So that the material or solid or food material is close to us and we can start a procedure, we can start a phagocytosis. So once the signal is relayed, other phagocytes pick it up. So signal is picked up by the other phagocytic cells too. And they start moving towards the material to be engulfed. They start moving towards the cells or towards the area where the food material is there. Right? So they follow the signal produced by one of the cell. So exact nature is not known, but it is presumed that there is some kind of a chemical reaction called as a chemotaxis between the cell and the particulate matter, right? That's why it has been detected by the cell, no? Just like in the pinocytosis, what is there? Some triggering points. As soon as the solid material comes in contact with the plasma membrane, some triggering point is there that start invagination, right? And triggering points are variable. Protein is acting as one of the good triggering point, right? And then that further leads to the pinocytosis. Right. So here in this case also some kind of a chemical reaction that which takes place between the phagocytic cells and the food material or a solid material that being released in the form of a signal, or it is been released by the phagocytic cells that picked up by other cells and they get aggregated towards that site where the food material is available and all together carry out the phagocytosis. Right, the same way happen in our body. If you got some injury, right? If you got some injury, so what happened? Slowly that fingers start swelling, and it become more and more reddish for 1 or 2 days or 3 days. What it means? It means that to the site or to that area, what happened? The blood cells, they have been dilated so as to force more amount of a blood vessels, they have been dilated so as to force some more amount of blood to that affected area, right? And why more amount of blood cells? Because more amount of phagocytic cells, that is W.B.Cs, they have been directed to that area, right? And more W.B.Cs, they get gathered and they pick up immediately, they feed on, or they capture all the foreign particles, that is bacteria or viruses, whatever is there, as well as worn out cells, dead cells if there. So they also feed on it. So it means if the cell, one of the cell detect the material, food material or foreign material, it is going to relay some kind of a signal, right? And that signal is picked up by other phagocytic cells and all together gathered and feed on, or you can say carry out the process of a phagocytosis. So the phagocytosis is also observed in the macrophages, or you can say in the WBCs as well, right? So dear students, in the present lecture, what we have studied the process of pinocytosis, how the pinosomes they form along with the examples. We also studied the process of phagocytosis, how phagocytosis takes place along with the example. Okay, and tomorrow we will, or in the next lecture, we will study the functions of cell membrane, different functions of cell membrane.